All right. Welcome, everybody, online and in person, to the final set of package demos for Bioconductor 2024. This one will be from Peter Wong with the Van Andel Institute Graduate School at Michigan State University. And I'll be demonstrating bamSliceR and how to fish for tandem duplication signatures on both controlled and non-controlled access genomic data.
Yeah, thanks for coming to the next section of the package demo. And this QR code you can scan if you want to access the Van for the demo. And I also will go over the Van. Yep.
You might want to embiggen.
Too big.
Small and a little bit. Oh, somewhere.
Does that look good?
Hurrah.
Okay.
No, it's not.
Okay, so today I'm going to introduce the bamSliceR package we have recently working on. And I want to mention that the motivation behind developing this package at first can trace back to a pediatric acute myeloid leukemia case that we see that patients didn't carry any seemingly risk factors to drive the disease, but these patients carry mutations on histone 3 genes that called S3K27M. These mutations has never been documented in the AML patient before at the moment, and but it's been well documented and in pediatric brain tumors, which it's highly prevalent in pediatric brain tumor, and also it has huge impact that can cause the global reduction of trimethylations on the 3K27 sites of the histone. So we started to ask the questions: what's the role of these mutations carried in this patient, and how prevalent this mutation might be? How prevalent this mutation will be in the AML patients. So we started to develop this package, hopefully to ease the process on analyzing the patients' sequencing data from remote database. And specifically, we focus on its application with the Genomic Data Commons, or GDC, which is hosted by NCI, and it's one of the major resources for cancer research. And also, it offered a vast collection of clinical and genomic data from patients from the large translational consortiums like TCG and Target with all type of cancer. And also, it provides high performance data, acquiring sharing infrastructure that allow the community to access the data. And among all the, I forgot to open the website. Among all the endpoints provided by GDC, this bamSlicing API probably is one of the most useful functionality that allow users to extract the reads from the sequencing data remotely by giving the genomic regions or the genes, specific genes. So, and further, this bioconductor package with the same name implement all those services to the R interface. And allow user to query the data from GDC that in the R environment directly. So we think this is a great tool to examine the variants from patients across the different cancer studies. And so we developed a bamSliceR to integrate all those functionality in it. So this is the general pipeline of the bamSliceR. So the first thing is that we implement this API to query to slice and download the data from GDC. And I forgot to mention that most of data on GDC is control access, and you need a token for this slicing step. And you can find more details in the links for how to set up the token on your local machine and how to get permission on this access data. And after downloading the data, we automate the process to tally the variants read counts from those BAM files, followed by annotations. Yep, we also automate the process on transcripts aware annotation. Unfortunately, GDC didn't provide the BAM slicing from GDC didn't allow us to slice the transcriptome BAM files. But by using the same tools, we see that's pretty easy to subset those transcriptome BAM given the context contains all the transcripts IDs you're interested in. Okay, so in the end, you can expect bamSliceR to give you all the matrix about recounts of reference and alternative alleles, and also most importantly, the variant allele frequency of a specific variants. And the end goal of the bamSliceR will be that we implement some suitable statistic framework to model this biological relevant event, like allele bias between the genomic DNA web and RNA web, and also the transcript bias between the genomic and transcriptomic RNA web. Yeah, and I also want to emphasize that except the first step, all our functionality can be used on your local BAM files. Yep. Okay. So let's go to the first step on how to identify the BAM files information you want to download with. So if you want to query more other types of data from GDC, you can go to the website for more information. Yeah. So we simplify all the querying process in this single function to gather the information of BAM files. All you need is these three things that specify the project ID and the experiment strategy, also the workflow, and we provide these three functions for you to find out the keyword about these three parameters. So as you can see, for example, here the available project on GDC, and also the available experiment strategy for BAM files usually will be RNA-seq, whole genome-seq, and whole exon-seq, and also the available workflow given the project ID and the experiment strategy. As you can see, for example, here in this target AML cohort, there's a 3,225 BAM files available for RNA-seq data. And GDC used the standardized protocol to align those reads. And for RNA, it used the STAR with 2-pass pipeline. And for DNA, it will be BWA. So, yeah, I think it's finished. So you will get a data frame, including all the information you need to download the BAM, like the ID and the file names and sample type. Yeah. So the next thing you need is to specify the genomic regions you interested in. So in this case, as an example, which I choose 19 epigenetic related genes associated with the AML. And we provide these get genes coordinates functions to easily extract the genomic ranges either in GR range object format or the vector of string format that are required by the bamSlicing. Yeah, I should run this first. Yeah, those are the gene names we choose as an example, and you can then run the function to get the genomic ranges. Yeah, it will look like this as a G ranges or vector of string, and then you're all set, and you can just you can just run this function with all the information you prepared previously to slice and download the BAM. So I want to show you the. I want to show the figure here. Like you can see that for extracting reads, for extracting reads from 2,000 plus object with 3,000 plus BAM files from sequencing data on these 90 genes, you will end up with only 62 gigabytes, which I think pretty manageable on your laptop. And when the original size would reach to 39 terabyte. Yeah. So, and if you're only interested in 1 or 2 genes that span 100,000 base pairs, you can probably just ignore the requirement for the space. Okay, so after downloading the BAM, we can start to tally in those recons from those BAM files. And again, you need to specify the genomic ranges and also specify the list of BAM file names. What I did here, just scan this text file contain all the names of the BAM. And further, in order to tally in the reads, you also need a GMAP genome object, which is also required for this GMAP package we're using to tally in the reads. So it's just the object contain the indexed genome sequence. So basically, you just need to provide the resource of your sequence, reference sequence, either in the FASTA file or a DNA string set object, and also could be a VS genome file. So here we are for tallying the genomic BAM, we're using this VS genome package from Backconductor. Contains SG38 full sequence, full genome reference sequence of human. And for the transcriptional BAM, you can choose the FASTA file that can be easily acquired on GenCode website. Yep. And then we're ready to tally the reads with these three parameters we prepared. And also, I want to mention that this tallying read step is one of the most time-consuming step. So if the machine you have can access multiple nodes, or you can access the HPC resource, we provide these three functions allow you to customize on how many nodes can parallel tallying the reads on genomic ranges, and how many nodes you want distributed to the BAM files, number of BAM files. Yep. So let's go back to the code here. So after tallying the reads, you can accept a VR ranges object. VR ranges object contains the information about variants, including the genomic coordinates and the base changes and all the recounts information. And then before the annotation, we want to save those recounts information to the vector column, because these VR range specific information will be lost after annotation. Yep. And also, we have a function to match back those variants with the BAM file information. Yep, as you can see here, you will add the date information in the as a extra columns in here. And then at this point, you are ready to, you can perform some simple filter based on the cutoff the allele frequency or the total read depth. Yep. Then we're ready to annotation. So we utilize the functionality we already set up in the bioconductor package called a variant annotation. Customize it a little bit, and so it's pretty easy to use this one function, and you will get the all the information about the consequence of the variants, including the amino acid changes and the type of the mutations, type of the variants. For example, here, yeah, you can see the amino acid change for each variant in documented, and also the consequence. So, and then we provide this optional annotation using the this terminal toolset called variant effect predictor. And we think that if you want more insights that related to the variants, and if you want to predict the potential impact of the variants, you can use these tools. So we have functions set up to convert the GR ranges to the VCF file that requires the input for this tool. Yeah, and you'll be all the unique variants right into the VCF file and to be run with the web. Yep, and after running the web, you will output another VCF file contains all the information of these variants. Then we can extract the consequence information from it. Then we will get the actual insights these variants, and then we use this function to match back those information to our GR ranges. Yeah, and also want to mention that some of these columns produced by the web is required for the a bioconductor package called MAPTool that can be used to analyze and visualize the annotate variants. I will show you an example very quick. So if you only care about the single nucleotide variants, then we're pretty much already done here. But if you also care about the small insertion or small deletions, then we need this extra step because the GMAP, when telling the reads, you will not record the reference recon for these insertion and deletions. So as you can see that if you filter out all the indels, you will see allele frequencies are equal to 1. We provide a function using the pi-up from the RSAM tools to we provide this fixed indel reference count function to that utilize the pi-up from RSAM tools to retally on those loci that with indel events. And you can see that with the same MAPQ and base quality parameter, you will get the same results of those recon from pi-up and tally reads. Yep. Okay. After annotation, we provide some functions to visualize those distribution of the WAF. For example, here, if you wanna check the multiple loci, variants on the multiple loci for single genes, you can choose the gene names and the loci, the position of the variants reported by the reported previously, and to see the bar plot represent the distribution of the WAF for each different allele. And also, if you can also use this function to plot a single allele on multiple genes. For example, the figure we show here is this single loci at 27 of the histone 3 protein that are coded by all these histone 3 genes. Yep. And if you want, you can totally skip that and using the map tools to do all the analysis, including the oncoplot or survival analysis, or do the mutually exclusive test. So we have a function set up to convert all the GR ranges, including those variants, to the map tools format. And basically, that's all the plots you can generate from the map tools, and there's a lot more than this. So I want to highlight that. So using this pipeline, we definitely find more patients carry the H3K27M mutations, and also characterize some mutually exclusive events of these mutations with other epigenetic mutations. So hopefully the example can give you an idea on facilitated by bamSliceR API and also all the package from the bioconductors, because it's we can carry this type of analyzing pretty fast. Yep. And next, I want to go over some useful functionality we implement recently when working with the transcriptome BAM. It's pretty similar, but with a little different effort. Oh, not this one. So as you can see in this slides, all the basic steps is almost the same when you tally and annotate those BAM files, genomic BAM files. But for the transcriptome BAM, you lost the information of the genomic coordinates, right? You only have you aligning those reads against the transcribe. So we need to prepare different. We need to produce different format of different information of the file that contain the transcribe coordinates information to be compatible when tally and annotate those transcriptome BAM. So for example, here we have this utility implement here to convert the original GFF file, which is the annotation file from can be accessed from the GeneCode website. We have functionality to convert all the genomic coordinates to the transcript coordinates. Yeah, we'll see example here very quick. And also, we also want to, for tallying the transcriptome BAM, we also want to find the transcribe that compatible with the variants. So we have this get disjoint overlap bean function to create these disjoint beans that contain all the transcribe overlapped in the beans with both the information about genomic and transcriptomic coordinates, and then use these beans to match all the transcribe of the variants. Yeah, and also we use this modified GFF file to make the customized TXDB object, which is compatible with the variant annotation package we used before for the genomic BAM. Yep. Let's go back to the live coding here. Yeah. So for this example, we using the data from Lucy genes, that we map those reads from extract from the genomic BAM and realign those reads to using the minimap to produce those transcriptome BAM against the reference transcript sequence that acquired from the GeneCode website. And for example, here after tallying the read, you will get this exactly same VR ranges object. The only difference is that the sequence names right now it's the ID of the transcribe instead of the chromosome name. And then in order to annotate those variants tallied from transcriptome BAM, we need to first prepare a modified GFF file that contains both the genomic and transcriptomic coordinates. And as you can see here, this the original GFF file. And we have this function here to calculate the Yep. There you go. Yep. You can see that now all the secret ID will be transcribe ID, and all the coordinates will be swapped to the coordinates based on the transcript. Yeah, and we also maintain the genomic coordinates that match to those transcribe coordinates. And I also want to mention that this function will maintain the hierarchy of the features. So the output of this GFR, you don't need to index it anymore. Yep. Okay, so for next steps, we think that the transcript that overlap with the variant but have few or no reads mapped to them might not be recorded and could be filtered out. Can be filtered out in the tallying steps, but we think that these cases are still informative for just questions such as why those mutated transcripts are not being expressed, whether the mutation will trigger the mechanism like nonsense media decay that will probably eliminate those transcripts. So in this case, we want to just recover those missing transcripts that are compatible with the variants. That's why we set up this find. That's why we set up this couple functions that allow us to create a bean that contains all the transcript overlap at these beans, and we will record for each beans. We'll record both the genomic and transcriptomic coordinates of it, and then for all those variants we tallied before, we will search it, find the overlap of all the beans with this variant, and check each beans that whether there's some alternative transcript that also compatible with the variants, and retally the recons for these transcripts. Yep. I can show you an example here how this function will looks like. So the example here contains one variant from one patient at this transcript, and for these same patients, we can find whether this. Wait. Oh, okay. That's what it looks like for these. Yeah, this John beans function would look like. And as you can see that for each beans, you will contain all the transcript overlap with it. Yep. And then with that information, when you call this function here for this function, let me see. Took a while. Yeah, as you can see, for this single variant of this patient, for this single patient, you can find all the other transcripts that are compatible with these variants based on the location of the variants. Yeah, and after all these steps, we can start to annotate those tallied variants for transcriptome BAM. So we set up this this wrapper function that including 2 steps to annotate transcriptome variants, and the first one is pretty much similar to the one we used to use in the genomic BAM files, and all we need to do is prepare this customized TXDB object from the gene code from the GFF file, from a modified GFF file that contains transcriptome coordinates, and also we need to provide this sequence resource as a faster file format that contains all the reference sequence of these transcripts. Yep, and we can run it. Yep, and just like before, we will have the amino acid change for those variants, like from here, and also it's the same information as before. Yep, and further, we also want to map the locus with the feature of each transcripts using the GFF. Using the GFF annotation file from gene code, which can be done pretty easily. Just use the modified GFF file. As you can see here, for each variant, you will get information about the feature of the variants according to the transcripts. And was also whether it's, for example, whether it's at the stop codon region or not. Yep. And to save time, because I said before these 2 functions are wrapped in this single function, and all you need to do is to just use this function to merge all the information of the annotation from different resources. Yep. Yep. Okay, so that's all the pipeline we use to tally and annotate either the genomic or transcriptome bands. So right now we have allele frequency information derived from different alignment strategies, and now the question will be how can we use this web information from them to somehow infer the actual transcript that carry the variants. Yep. And you can back to the live code. You can see that we set up the function to merge. We set up a function to merge to match the web for variants from either the genomic band and the transcriptional band. As you can see, an example of this variant here. In this data frame, it contains the web from the genomic band, which should be the same for these patients, and also it contains the transcribed allele frequency for each transcribe here. Yeah, and you have all the information of the coordinates for these variants. And so we are still working on this statistic framework to how to use these two values and the recons to model this like which transcribe actually carry the variants. We think that rather than pretending that all of the evidence has equal weight, we can probably instead test for the null hypothesis that each compatible transcribe has a variant allele frequency that generated from the same underlying distribution sampled from the genomic web, and with the total depth number of phonetic trials. So then our prior will be the allele frequency of the variants on transcribe X equal to the allele frequency of that allele of the genomic web. Yeah, and so when the genomic web will follow the beta distribution, the alternative depth of the transcribe will follow a binomial distribution. So, and then we can probably do a Bayesian model comparison test to see to test the alternative hypothesis that at least some of the transcribe has the different allele frequency with the genomic allele frequency. Yeah, and also we also think that those reads that support the evidence for specific variants will have different weighted on the evidence, because based on the degree of ambiguous mapping for each transcribe. So we set up a function that can extract the renames given a specific variance and tabulate the base change for which renames support either allele frequency, allele base, a reference allele base, or the alternative allele base. As you can see here, let me run this code very quick. So yeah. For example, for this variant show here, we will go back to the BAM files and document all the renames that supports either the reference allele or an alternative allele. So by getting this information, we can probably weighted the evidence based on the output from this function. Yeah, I think that's all. The implementation we finished so far, and we will be keeping working on this to provide a more accurate statistic test to hopefully infer some interest biologically relevant questions. Yep. And yep. And thanks team for all the great ideas. I feel that some of them I haven't make it real right now, but I will keep working on that. And thanks Laura on contribution of some of the code and also testing the package on her Streamline project. And also thanks Eve, our lab manager, help me prepare the presentation. And thanks also for Amber help me prepare the presentation. And thanks Sean for some of the advice to improve the package. I think it would be much worse than if without Sean's help. And also thanks Alex, help me put extra effort to put this demo on live. And thank Eric who organized all this schedule. Thanks.
So that was our last session. We do have a closing session in the auditorium. It will be very short. So if everybody could head over there now, then we will call wrap on 2024.